And then I open this academic ceremony in which Min Deng will defend the academic thesis called Inflammation and MYOSTEATOSIS in Pancreatic Cancer CACHEXIA. That's all, CACHEXIA. Before we go there, I would like to introduce some of the people here. First of all, of course, your supervisory team, Professor Olde Damink and Dr. Rensen, who have been very, very helpful, I hope, in preparing the thesis. And of course, you're being supported by Annette Duivenvoorden and Wenbo Wu, who are the paronyms, as we call them. My name is Nando de Vries. I'm replacing director here, which just means I'm master of ceremonies. May I now invite you to give a summary of your work?
Yes, thank you, Proctor. Dear Proctor, opponents, supervisors, family, friends, and colleagues, and all those joining online, thank you very much for being here today. In the upcoming 15 minutes, I would like to share with you the summary and main findings of my thesis, titled Inflammation and MYOSTEATOSIS in Pancreatic Cancer CACHEXIA. Pancreatic cancer is a significant global health concern, ranking as 12th most common cancer worldwide, according to the World Health Organization. In 2020, it accounted for about 500,000 cancer new cases and led to about 500,000 deaths globally. The 5-year survival rate for pancreatic cancer patients remains low at 12%. Remarkably, about 80% of pancreatic cancer patients develop CACHEXIA during the disease course. So, what's cancer CACHEXIA? Cancer CACHEXIA is a complex syndrome characterized by significant body weight loss, including the loss of skeletal muscle mass and fat tissue, and which cannot be reversed by nutritional supplementation. Cancer CACHEXIA, this disease affects up to 80% of patients with advanced cancer, including pancreatic cancer, gastric cancer, lung cancer, as well as colon cancer. Among these cancers, patients with pancreatic cancer have the highest prevalence of CACHEXIA, experiencing body weight loss about 40% within 6 months. And more than 50% cancer-related deaths can be attributed to cancer CACHEXIA. To date, the mechanisms underlying cancer CACHEXIA are still not fully understood. It's believed that circulating factors rising from host, tumor, and immune cell play a key role during the development of cancer CACHEXIA. However, the characterization of immune system is still remain to uncover. Therefore, this thesis begins by examining innate immune system in pancreatic cancer patients, specifically focusing on complement system and neutrophils in relation to cancer CACHEXIA. Complement system is an ancient defense system that plays a virtual role in defending against pathogenesis. It can be activated through 3 pathways, as you can see in the picture: classic pathway, lectin pathway, as well as alternative pathway. These pathways differ in their initiation and produce a key enzyme called C3 convertase. Upon activation, complement system can promote phagocytosis, cell death, as well as inflammation. Considering that systemic inflammation is the hallmark of cancer CACHEXIA, we explored whether the complement system is activated in patients with or without cancer CACHEXIA. To investigate this, we collected blood samples from pancreatic cancer patients with and without CACHEXIA and measure complementary factors such as C1q, MBL, C3A, and TCC. As expected, the key complement system activation effector C3A was significantly higher in cachectic patients with inflammation as compared to those patients without cachexia. Additionally, the level of C3A was positively correlated with the concentration of its upstream Schwann-Q and downstream TCC. These results suggested that classic pathway may play a role in complement system activation in pancreatic cancer patients with cachexia. Another player of immune system we studied is neutrophils. They are known as their ability to migrate to site of infection where they can release virus of protein to initiate and amplify the pro-inflammatory process. Upon activation, neutrophils can release elastase, calprotectin, as well as lipocalin-2, the latter of which recently has been shown to involve in cancer cachexia by suppressing food intake in mouse model of cancer cachexia. Whether the same mechanisms applies in human patients is still unclear. Therefore, we collected blood sample from pancreatic cancer patients with and without cachexia, and we measure circulation lipocalin-2 level as well as inflammation. We also record patients' appetite. In this case, it refers to food intake. While cachexia patients tended to have higher circulating lipocalin-2 levels as compared to those patients without cachexia, the significance is not different. Furthermore, there was no significant difference in circulating lipocalin-2 level in patients with normal food intake and patients with reduced food intake. So these results do not support the notion that circulating lipocalin-2 contributes to cancer cachexia by suppressing patients' appetites. Next, we also examined the neutrophil activation marker in pancreatic cancer patients with and without cachexia. Again, although we see cachectic patients tended to have higher neutrophil activation markers as compared to those patients without cachexia, the difference was not different. However, when we subdivided the cachectic patients based on their circulating lipocalin-2 level, we observed the neutrophil activation in cachectic patients with higher lipocalin-2 level. Furthermore, there was the strong correlation between circulating lipocalin-2 level and neutrophil activation markers as well as the inflammation. It's also worth to notion neutrophil activation in pancreatic cancer patients also significantly correlated with the complement system activation. Considering that complement system activation and neutrophil activation both have been linked with inflammation, there exists a potential positive feedback loop connecting complement system activation, neutrophil activation, as well as inflammation. The inflammation rise from tumor cell and immune cell also has been shown to involve in muscle wasting. It's another key feature of cancer cachexia. So the muscle wasting can refer to lower muscle mass as well as poor muscle quality. In the field of cancer cachexia, low muscle mass has been extremely explored. However, the poor muscle quality, also called MYOSTEATOSIS, gets less attention. Therefore, the second part of the thesis characterized the myostetosis in pancreatic cancer patient and aiming to study the underlying mechanisms. So, what's myostetosis? Myostetosis refers to abnormal fat accumulation in skeletal muscle. As illustrated in the picture, you do see a lot of fats indicated in yellow in the myostetosis patient as compared to the normal patient. This condition has been linked to muscle weakness as well as reduced survival rates. In pancreatic cancer patients, our results showed cachexic patients had significant higher fat accumulation in their skeletal muscle as compared to those without cachexia. And the fat accumulation in skeletal muscle was significantly correlated with body weight loss and inflammation. These two are the key features of cancer cachexia. Additionally, by using mass spectrum imaging, we observe the histology spatial distribution of fats. Here is an example. As you can see, the cachexic patient had abundant expression of the least fat and sparingly expressed in no cachexic patient. So in this case, more yellow means more fat accumulation in the skeletal muscle. We next ask what's the mechanism behind myostetosis. Previous study already showed tumor-derived factors and inflammation play a role in the development of cancer cachexia. These factors can induce breakdown of fat tissue, leading an increase of fat in bloodstream. There may result more fat uptake by skeletal muscle. On the other hand, these factors can disrupt fat burning in skeletal muscle. Considering these findings, we hypothesize the tumor-derived factors can promote fat accumulation in skeletal muscle. To test this hypothesis, we collected tumor-derived factors from cachexic patient or non-cachexic patient. Then we exposure the tumor-derived factors to myotube and monitor the lipids fat accumulation in myotube. As expected, we do see significant higher fat accumulation in myotube for the tumor factors derived from cachexic patient as compared to those tumor factors derived from no cachexic patient or no tumor factors. As mentioned earlier, fat accumulation in skeletal muscle can caused by an imbalance between fat uptake and fat breakdown, involving more fat uptake or less fat breakdown, or both. Indeed, in our study, we do observe an upregulation of gene coding for fat uptake and a downregulation of gene coding for fat breakdown. So to wrap it up, the correlation between complement system, neutrophils, and various aspects of cancer cachexia were studied. We show complement system activation, neutrophil activation in pancreatic cancer patients with cachexia. Additionally, we reported fat accumulation in skeletal muscle of cachexic patients. This may be caused by tumor-derived factors. Overall, the thesis list finders underscore the wide role of inflammation and tumor-derived factors in the development of cancer cachexia. Thank you very much. Now I give the word back to Prorector.
Who will thank you for this presentation. Thank you. So now we'll proceed to the actual defense of the thesis, and the first question will be asked by Professor Dirk de Ruijser. He's a professor of radiotherapy, in particularly respiratory oncology at our own Maastricht University. Professor De Huysse.
Thank you very much. First of all, I want to congratulate you, your whole team, and of course your family with the fantastic work you've done. But I'm here to ask some questions, of course, and I want to ask one of your paronyms to read proposition number 6.
To treat pancreatic cancer more effectively, we should prioritize anti-cachexia treatment over targeting the tumor.
Thank you. Now, basically, the proposition number 6 suggests that you should treat cachexia first before treating the tumor. But throughout your thesis and your whole hypothesis, I have the impression that the cause of cachexia is cancer. So why wouldn't you prioritize to treat the tumor instead of treating the consequence of the tumor, that is cachexia?
Highly esteemed opponents, thank you for your question. The main reason we thought we should treat cachexia first before treat cancer, because for most of cachexia patients, as mentioned in this thesis, most of them cannot suffer or cannot get like chemotherapy already because it's so toxic for them. So in principle, it would be kind to first treat cachexia. When they get like the bad condition, then we can treat the cancer. So that's why we thought maybe we should first treat cachexia instead of tumor.
But isn't this is this also true for pancreatic cancer? I mean, I'm working in the lung cancer field, but we see it also in lung cancer in a lot of fast-growing tumors, that it's a kind of catch-22. If you wait too long, then actually the patients will be dead already. So how does this is applicable in typically pancreatic cancer patients, which is the subject of your thesis?
Yeah, if I understand your question correctly, you mean in the colon cancer, it's kind of the same case, the patient will be die fast. Then, so what's your question again?
My question is actually, if you first treat treat cachexia, and actually, as far as I know, we don't have good means to treat cachexia, isn't this a problem that you will treat the symptom rather than the cause, and hence, at the very end of the day, there's no big gain, or do I see it wrongly?
Yeah, I think you're right, because most of pancreatic cancer, they like pass away quite fast. So that's the reason.
And then from all the work you have done, could you identify the kind of threshold where the patient comes in a kind of refractory situation where the whole cachexia process is irreversible, and where you would say these patients are really, sorry to say so, the only thing what we can offer to them realistically is best supportive care? Where is actually the threshold? Do you have some biomarkers where you could, in which you can objectively identify patients who come in a refractory cachectic state?
Yeah, that's a good question.
Coming from your thesis, huh?
Yeah, based on my thesis, it's hard to tell because so far the biomarker, as one of the slides, is show there potential some markers related to cancer cachexia. However, targeted single mediator, it seems works in mouse model. However, it's never been worked in the human patients. So my answer is so far there's no effective like factors that play a key role during the development of the cancer cachexia. But in this thesis, we do see inflammation and myostetosis may play a role. So for the clinic, what could we do? I think maybe we can take into account for body composition analysis to look at if the patient with or without myostetosis, because we know the patient with myostetosis, they get like the reduced survival as compared to those without myostetosis. So that's my answer. Thank you.
And if your promoter would give you a real big grant, something like 10 million euros or so, and you may do whatever you want for 5 years, how would you then design a trial or something else in order to come with what I, the clinician, could use to identify the high-risk patients? How would you, and there's no financial limit, huh?
Yeah, that's...
How would you design it? What would you do?
Yeah, based on my research, what would I do? I will focus on two aspects. The first aspect I would do to just based on CT scan, because most of patients, for the diagnosis, they will take CT scans. So actually it won't be cost additional financial. So what we can do, we can just simply extract the L3 CT scan, then we perform body composition analysis. Then for doctor, you will know this patient is myostetosis or not. So at least the doctor, they know, they potentially know the patient will survive long or not. And the second part, what would I do? I will still investigate how the inflammatory factors such as IL-6 and TNF-alpha, these pro-inflammatory factors, how these factors contribute to cancer cachexia, either induce more fat accumulation in skeletal muscle or just induce muscle atrophy. So I will just spend money on that to specifically look at the mechanism underlying cancer cachexia, specifically focus on muscle, because it's the key feature of cancer cachexia.
Thank you very much.
Yeah.
I give the word back to Mr. Pro-Rector.
Yeah.
Thank you, Professor de Ruysen. I now turn to a guest of another university, and we're always very proud when there's opponents coming from other universities, because that attests to the importance of the work, because otherwise they would not do the effort, of course. So I welcome Dr. Maria Room, and she is the group leader of the Cancer Cachexia group at the Institute for Diabetes and Cancer of the Helmholtz Institute in Munich, in Germany. Your question, please.
Hello. Congratulations, first of all, to your very clear presentation and to the great results that you presented here today. I hope you can hear me okay. Maybe give me a brief nod if you do. Okay, good. So I would like to get your opinion based on everything you read and also your own results. What would be the most important factor contributing to cachexia?
If let me guess, I would say information, because based on my thesis, since everything linked to information, although previous study already identified certain of potential biomarkers such as IL-6, IL-8, however, these factors, yeah, as mentioned, is not effective for the treatments. So if they also, there's one factor I haven't mentioned is the GDF15. It's quite a hard topic since you also work in the field of cancer cachexia. You may also read a lot about this. GDF15 play a role to regulate food intake in patients, because we know cachexia patient also had anorexia. That means reduce the food intake. So I would say, in one hand, pro-inflammatory factors play a role, but also some factors related to food intake, such as the GDF15, lipocalin-2. These factors may also need to pay attention on that. Especially in our thesis, we initially resort to look at lipocalin-2, how it contributes to regulates food intake in human, because in mouse model it shows lipocalin-2 can inhibit food intake therefore contribute to cancer cachexia, but it seems not a case in our study. So I think maybe there are more studies need to follow up. Yeah, thank you.
Okay. Then yes, thank you for your answer. Let me challenge you on this a little bit, because you say inflammation is very important, and it's actually a hallmark of cachexia, and it's depending on the definition that you use for classification of cachexia, it's one of the factors that contributes to the characterization or to the classification of a cachectic patient. So I found it quite unusual that then you divide your groups, your patient groups, in cachectic and cachectic with inflammation. Can you please comment on this? Like, what makes the cachectic non-inflammatory group cachectic, or why is it not important in that group then?
You mean why I divide group into cachexia with and without inflammation? Yeah, thanks for your question, esteemed opponents. Why we take into account to group patient based on their systemic information? In our case, we use the C-reactive protein because it's commonly used in clinical research, or just for clinic, because it's commonly used to determine information, systemic information level. And back to your question, why we also take into account information, because based on previous study, we thought information play a role, and we also investigated the innate immune system, because this information can released by immune cells such as the neutrophil, macrophage. So we think if we want to study innate immune system, we may also take into account the level of information, inflammatory factors. Yeah, thanks.
Okay, so I was just curious, because then the patients without inflammation still have cachexia, so other mechanisms must drive the cachexia in that subgroup. So would it make sense on the long term to classify, say, different cachexia subtypes and to treat them differently as well?
Yeah, I agree. So you mean if we can treat patients based on their information, if I understand correctly? Yeah, I think it is quite important to characterize patients' information states, because based on this thesis, most of patients with higher information, they actually get more fat accumulation in their skeletal muscle. Also, with higher information, they got neutrophil activation as well as complement system activation. Certain level of complement system activation and neutrophil activation may help to anti-tumor progression, but if it's too high, it may play another role to promote tumor genesis. So definitely, I think we should also take into account, take the information level states into account for patients' treatment. Thank you.
Then one last question, perhaps. Are these lipids that you observe in the muscle, is there only myosteatosis, or is it a general theme that you have more lipids in different tissues, say in the liver as well? So would it be an overarching mechanism? And if it is, then what do these lipids do to contribute to disease?
Yeah, thanks for your question. We sought to also look at the mechanism, what's the lipids comes from. So we thought might be in lipids accumulation in skeletal muscle, might be the lipids comes from muscle itself, or just uptake from the other organs, the degradation of other organs such as adipose tissue and also liver tissue. Actually, I didn't get time to look at lipids profile in liver and adipose tissue, but that definitely really interesting to look at whether the lipids accumulation in skeletal muscle comes from the lipolysis of adipose tissue or comes from just from the liver due to the acute protein, acute phase protein. Yeah, thank you.
To return.
Yeah, thank you very much for your contribution. And I turn to another guest, and it's Dr. Luca Silo. And she's an associate professor of molecular metabolism in cancer and aging at the Department of Biomedical Sciences, University of Copenhagen, which of course is in Denmark. So welcome to you as well. And the floor is yours.
Thank you very much, Mr. Prorector. And congratulations, Mr. Candidate, on all of your hard work. It's very nice to see you again. I will pick up a little bit where Maria Rome left. So can you, in brief, outline what we know about the molecular mechanisms that drive myosteatosis?
Esteemed opponents, thanks for your question. For the mechanism drivers to myosteatosis, yeah, I read some literatures. Also, one of yours, I think it might be the insulin resistance play a key role, because in my thesis we found like PI3K AKT pathway is activated, and we know this pathway play a key role in insulin resistance. So there might be the activation of AKT pathway promotes more fats uptake or lipids uptake by the muscle cell. On the other hand, this activation could also the sign of stress corrective factors such as the PCG1. It's also upregulated in cachexia skeletal muscle. So these factors can also contribute to mitochondrial biogenesis. So in our case, we see the mitochondrial biogenesis is breakdown. So I would say the mechanism could be mainly caused by mitochondrial dysfunction and also the insulin resistance. Thank you.
Thank you very much for that answer. And with that in mind, I would like to take us into a little bit of a dive into your pancreatic organoid system, combined with the skeletal muscle tissue culture experiments that you have done. So can you tell us a bit about the strengths of this system, but also maybe the limitation of this system with all of these different mechanisms that occurs in vivo in mind?
Yeah, for the organoids parts, we discussed a lot with my supervisors about the key limitation I thought might be the standardization of two developments organoid system, because you can see the same amount of the standard cell to establish organoids. However, you could not control the size of two organoids and the numbers of organoids. Also, it's really hard to look at the shape change over time if you do treatments. So this is the main limitation, I would say, to standardize. How can we control to establish the organoids? Because if the numbers or the size of organoids is not the same, then your data you corrected might be not contributed by the tumor-derived factors. It's just because you load different amount of organoids. So that's the key limitation. Yeah, thank you.
Thank you. And so in your system, you have incubated media from the organoids, cell cultures onto muscle. And you saw this nice accumulation of lipids However, did the myotubes recapitulate other features of cachexia?
Sorry, could you repeat again? Thanks.
Sorry, I'm wondering about whether the system of the muscle recapitulates the cachectic phenotype, because you saw this accumulation of lipids. But apart from that, did you also see other markers of cachexia in this system?
Actually, we actually did morphology analysis, and we look at the marker related to muscle atrophy, such as the muscle ring finger 1, and HG1. We also look at the mitochondria, the shape of mitochondria, and we also look at apoptosis. But however, we only see lipid accumulation, and the gene related to lipid metabolism were affected. But for apoptosis, we didn't see any significant difference in myotube after treat with the tumor-derived factors from cachectic organoids or non-cachectic organoids. We also didn't observe the size of myotube was affected during the treatment. So no, we didn't. We do look at the other markers related to cancer cachexia, but most of them did not change between the cachexia and the non-cachexia. Yeah, but of course I have to acknowledge that the concentration of condition median, we diluted the condition median, but we diluted like 50%. It's commonly used from the literature, but I would suggest to maybe if I do like follow-up study, I may use the concentrated or use the condition median correctly from the organoids instead of to dilute it. Yeah, thank you.
Thank you for that answer. So did you measure any known tumor-secreted cytokines in your media and related that to the abundance in the plasma from patients?
Yeah, actually we found CAP, like IL-6, IL-8 is quite highly expressed in cachectic condition median, let's put like that, as compared to the condition median correct from non-cachectic organoids. So that's quite interesting. Another one is the macrophage inhibitor factor. It's also was significant difference between cachectic organoids and non-cachectic organoids. So one of our follow-up study, we are looking at how these factors contribute to mitochondrial polarization, and we get some nice data. So these factors could play a key role as well. Yeah, thank you.
Thank you. So my last question is, I'm wondering why you chose to use C2C12 myotubes, because you have a human organoid cancer system, and then you put those secreted factors onto a mouse system. So I'm wondering why you chose this, and also what could be concerns about using a human system and adding it to the murine system.
Yeah, thank you for your question. That's a really nice question, because I also thought about that, because yeah, you're right, we collected condition median from human organoids. Why don't I use the human-derived myotube to test the effect? The main reason is that at that time, I think I only got like the C2C12, and it's also common used in the literature. I saw people from the US, also European, they use a lot C2C12 cell. I think this might be, it's just more common use, but it doesn't mean it's a good option to use that. But for investigates or explore more mechanism aspects, I would say it's good to use the human-based myotube. Yes, thank you.
Yeah.
And thank.
That was my question. Now go back to Mr. Prorector.
Yes, thank you very much. The opposition will be continued by Dr. Ramon Langen, and he's an associate professor in the Department of Respiratory Medicine of our own university.
Thank you, Mr. Prorector. Dear candidate, first of all, also my congratulations to the completion of your thesis and the elegant work that is described in it. I would also, of course, like to extend these congratulations to your promoter and co-promoter. And I would like to start my opposition with focusing on the work that you describe in chapter 3. In that chapter, you investigate the hypothesis that elevated plasma levels of lipocalin-2 cause cachexia, and they do so by reducing food intake. And then in the discussion of your thesis, you come back to these results and you actually conclude that neutrophil-released lipocalin-2 may contribute to cachexia, and that it may happen through affecting the nutritional status. And based on your work, I do not fully understand that conclusion, and I was wondering if you can help explaining that a little bit more, maybe starting by focusing on the first part of that conclusion, that there is a role for neutrophil-released lipocalin-2 in the induction of cachexia.
Yeah, esteemed opponents, thank you for your question. Yeah, the conclusion why we draw like that, because in page 30, in page 33, in this page, although we didn't find a significant difference in lipocalin-2 between cachexic patient and no cachexic patient. However, we do see the patient with malnutrition, the poor nutrition, patient with the poor nutrition states with the higher systemic lipocalin-2 levels. So although we did not dive into the mechanism why, but we do saw there's the increases in lipocalin-2 level in the patient with poor nutrition state. So that's the why we conclude lipocalin-2 may also contribute to cancer cachexia by influence patients' nutrition states. So that's the main reason why I drew this conclusion.
Yeah.
But I agree it should be put more effort to look at the mechanisms underlying this.
Yeah, I would also really like to go into those mechanisms with you as well. But first, there is of course another study in which they did find this association between cachexia and increased lipocalin-2 levels. And I was wondering if you can speculate on a potential explanation why their findings are different from yours. Does it have anything to do with the methodology, you think, or the cohort of patients that was investigated?
Yeah, thanks for your question. Actually, the lipocalin-2 paper, when we sent out the first time, we also, some reviewers say why your result is different from others. So the main reason we thought might because we investigate innate immune cell. This is really easy to get activation. So the process of the blood is really important. If the cell gets activation during the process, then you definitely get higher lipocalin-2 levels. Because in our study, actually we observed quite lower lipocalin-2 even in cachexia patient. But for the other study, they do observe the much higher lipocalin-2 level. So, but it's hard to say it's due to the process or just due to the reagents or ELISA kits. So I really hard to tell what's the reason. But recently I also read one paper. They mentioned lipocalin-2 could released by immune cells, such as the neutrophils is the key contribution of lipocalin-2. It can be induced muscle wasting as well as the fat wasting in a mouse model. So yeah, that's quite interesting to look at what's going on in the muscle and fat
So that would be a route independent of food intake that you are
Yes.
proposing by direct effects of lipocalin on those tissues. Yeah, I actually think that's quite an
Yeah.
interesting thought. And I think you also show data that indicate that there is a reduced amount of fat tissue actually present in people, in the patients with increased lipocalin-2.
Yes.
So that could support some sort of mechanism, at least a relationship between these two findings. Could you speculate on how that would work?
Yeah, in this thesis, I agree. We do saw a negative correlation between circulating lipocalin-2 level and satchel, that means the subcutaneous fat.
Yeah,
We
exactly.
do see like higher lipocalin-2 level, the less subcutaneous fats. So the reason could be I think the mechanism behind the association between lipocalin-2 and reduce the fat tissue could be the immune cell infiltrated in fat tissue, therefore they release lipocalin-2 or other protein acids that drive fats, lipolysis of fats or browning of fats. But it would be interesting to look at the marker of lipolysis of fat, also browning fat, because those two can cause fat loss.
Yeah.
Yeah.
I think that's a very attractive hypothesis to follow up on if you were to receive the 10 million euros that were already proposed.
Yeah, thanks.
But how would you approach that? How would you? What would be the first thing that you would do in order to build a foundation for the idea that lipocalin-2 might be directly acting on adipose tissue?
The first thing, of course, to correct fat tissue from patients, then we can look at the marker of, or to do like immunochemistry staining to look at this marker like lipocalin-2, because if you don't see higher level of lipocalin-2, then that may not support your idea. And if we do see the higher lipocalin-2 in fat from CACHEXIA patient, then we can do some gain and loss function experiments to see how these factors play a role in fat loss.
Okay. And what would be the loss experiment? What would you delete?
Yeah, could be use the RNAi to simply knock down the key receptor of the lipocalin-2 in neutrophil, or we can simply systemically knock down lipocalin-2 in we can systemically knock down lipocalin-2 in neutrophil in mouse model.
Yeah, or in the fat tissue like you proposed in the first approach.
Yes.
I fully agree, and I think that would be very exciting follow-up work, and hopefully there will be a way to actually accomplish this. So I'm very satisfied with your answers.
Thank you.
And I would like to return the word to the prorector.
Thank you very much, and I'm still looking for the 10 million. We now turn to Professor Matthijs Hesselink, and he is the Chair of Physical Activity and Metabolic Health in the Department of Nutrition and Movement Sciences of our university.
Thank you, Prorector. I also like to express my congratulations to the candidate and the supervisory team. I think we face a very nice thesis here that encompasses lots of different technologies being used, a wide array of approaches, which I think adds to the quality of the answers, but also sometimes adds to the inconsistency of data. And that's something that you have to face and to deal with. And related to that, or partly related to that, I will ask a few questions on chapter 4 and 5, I think. So I would like to go with you on a statement that you make rather frequently throughout your thesis, and that relates to, it's basically a reference to literature, I think, that circulating sphingolipids like ceramides, that they contribute to muscle wasting. And then my first question would be, why? How? Do you have any idea why circulatory ceramides can add to wasting, or where do they originate from? Can you tell me something about this?
Highly esteemed opponent, thank you for your question. For the ceramides, I think the main, how the ceramides contribute to muscle wasting, the it can be, the ceramides is known to induce lipotoxicity in cells. So that could be the main mechanism how ceramides contribute to muscle wasting. Also, one of the opponents, Dr. Maria, she's already published one paper. It shows certain of ceramides actually is kind of biomarker in cancer cachexia, which cause muscle wasting.
With it being a biomarker in the circulation, then the question always pops up, how do these ceramides get into the circulation? So where do they originate from? And if they have a role in muscle wasting, how do they get into the muscle? Is that known?
So far, I can say is most of lipids or free fatty acid comes from adipose tissue, but also might be also the dead cell that could be also a source, but mainly from adipose tissue.
Okay.
But we did not look at the circulating fatty acid profile in cachexia patient. So
then they come from the adipose tissue. I don't think they will leave the adipose tissue as ceramides, but okay, leave alone that. In your study, you will find, or you do find actually, based upon LCM SMS data, I think, elevated ceramides in muscle tissue of your cachectic patients.
Yeah.
If you do the MALDI approach, I'm not sure if you see the same thing.
Yeah, we did MALDI.
Sorry?
We did MALDI.
Yeah, okay. And there you see the elevation of the ceramides.
No, because the elevation of ceramides we observed actually comes from the LC-mass instead of the MALDI. Because MALDI, why we use MALDI? Because we also wondering how the ceramides distribution or other lipids associated with the cachexia, how they distribution. However, by using MALDI mass spectral imaging, we could not identify the ceramides. The reason could be the level of ceramides is too low to detect. So,
but I could also think of another reason. So if I look at page 97, figure 3, where you do the oil red O staining in your muscle sections, you actually do see that with a bit of a higher magnification, most of the lipids are nearby or in vicinity of the sarcolemma, the muscle cell membrane. When you move to the MALDI approach, you need to stick to a poorer spatial resolution, that's inherent of the technology. You do segmentation there, and if I understood correctly from the segmentation, you focus on the area that is within the muscle cell, but that would also imply that you sort of leave out the oil red O-based droplets that you see in vicinity of the muscle cell membrane. So if you were to use the inverted approach, not just what you see within the muscle cell, but without the muscle cell, in your same MALDI data, would you get the same profile? Did you happen to try that?
Would be nice to follow up, but no, we did not look at that yet.
Okay.
Yeah, thank you.
Proceed? Yeah. Okay.
But
so
we do have data. We can look at the lipids in between the.
As far as I understand, you would still have the data and could do the reverse. So that's a suggestion. Another thing that you could look into is that I think in the region of 1400 mZ or so, we notoriously find in similar data obtained together with the group from Ron Heren, cardiolipin, which is a mitochondrial inner membrane marker. Did you look at that?
No, but it would be really interesting to look at that marker. Yeah, we did not look at the marker yet.
Okay, so there's still a couple of things that that could be done together, or maybe just the two of you or whoever. Note that we still have data from skeletal muscle from control individuals, and that's my last question. You see there's some spatial resolution, some spatial differences within the muscle that you examined. You examined rectus abdominis muscle. We examined vastus lateralis muscle, which has a difference in fiber typing. Is the rectus femoris, is that more homogeneous in fiber typing? Or could the spatial difference that you see originate from different muscle fiber types?
At this time, we can do that, but we did not look at the muscle fiber type. But we also mentioned that in this thesis, it would be nice the follow-up study to look at the muscle fiber type, because we know in muscle there are like the different type of muscle fiber. Fiber type 1 may be related to oxidation, and the other one related to non-oxidation. But with the Professor Ron group, they are able to combine mass spectral imaging with the immunochemistry staining. So that definitely can be done by using the ArtStates technology.
Another reason to stay in touch. Thank you for your answers, and I give the word back to the prorector.
And I'm still looking for the 10 million, actually. You too, yeah, sure. Probably the final opponent is Professor Wim van Gemert, and he's a professor of pediatric surgery at our university.
Thanks a lot. Esteemed candidate, also my congratulations to you and your supervisory team for an impressive work, what you accomplished. You will like my question, which will be the last, I think, because I saw an acknowledgement that you like cats. And this is about cats. It's about actually famous, maybe legendary even, theory and slide from my own old mentor, Professor Soeters, professor in metabolism and nutrition. And he showed a slide which was referred to as the cat with the paw, or the sad cat with the paw. And in that slide you see a lioness with an injured paw, broken. And what she does is she will find shelter in a protected area under a bush. She will lie down and do nothing else. She will eat almost nothing, only what her companions from the troop will bring, and waiting to heal. What happens is that it will lose a lot of muscle mass. So endogenous proteins will be broken down and then synthesized to the necessary components to heal. So host defense, like cytokines, mediators, and things like that. So it's actually a beneficial and necessary process. The thing is that the fracture must be healed before muscle mass is completely depleted and the animal will die. In a cancer, you have this tumor, and host defense will not be able to completely heal. So the process will go on, and if nothing happens, muscle mass will deplete and will be significantly contribute to death, actually. So my question is, if you find a way to block this cascade for cachexia, what would be the impact on the treatment of pancreatic cancer in this case? And you can think of healing, survival, complications, quality of life, things like that.
Yeah, highly estimated opponents. Thanks for your question. Yeah, if we are able to break down the cascades of like muscle breakdown, definitely it would be benefits for patients with pancreatic cancer, because most of them are like in the UCI. So if they get the strong muscle, they definitely can go outside. So on one hand, at least they can have the best life. On the other hand, if we can block muscle breakdown, it's also benefits for the treatment, because most of cachexia patients they not handle chemotherapy or chemotherapy or other type of the immunotherapy. So if they get the good muscle, so probably they will hand over the process of the chemotherapy.
Okay. Yeah. Could there be also disadvantages because it also is important for host defense? So if you block that or block it too soon, could there be disadvantages like accelerated tumor growth, metastasis, things like that?
I never thought that. Yeah, that could be. Yeah, if you break down breakdown of muscle, could be also not benefits for patient in this aspect. Yeah. So, but I never saw that. Could be. Yeah.
Okay.
Yeah.
Well, what I think myself is that the process is beneficial, but if it gets too bad or doesn't stop because the tumor is still there, it will be disadvantaged because you cannot live with no muscle mass, and also your quality of life will be bad. But if you have treatments, you already said that, like surgery or chemotherapy, you will have a big chance of having complications or even not survive a treatment, intensive treatment like that.
Yeah.
So what I would think is that maybe it would be better to find a way to improve protein synthesis and depositing that in muscle mass, so that synthesis will be equal, or almost equal to breakdown.
Yeah.
So that you will still have enough muscle mass for your host defense mechanism and don't die of cachexia.
Yeah, I agree. Yeah, if we can like increase the protein uptake by muscle or other organs, might be get the similar. Yeah, thank you.
Thank you too for your answer.
Did you finish that sentence? Otherwise you might finish the sentence.
Yeah. Okay.
You did?
Yeah.
Okay. Well, then I will announce that the time appointed for the defense of your thesis has now passed, and the degree committee here will withdraw and discuss the quality of the thesis and of your defense. I would ask you and your company to await the results of these deliberations in this room, and thereby I adjourn.
Thank you.
The PhD defense has now ended. The degree committee will debate the candidate's performance behind closed doors. This process usually takes about 10 minutes.
Southside, 10 miles in my rearview mirror. I know what it felt like. My goals only getting clearer. East side to the west side. No place like home. If it's questions that you've got, go the extra mile and die. All your time, 'cause I'll go the extra mile. Long road to the south side, 10 miles in my rearview mirror. I know what it felt like. My goals only getting clearer. East side to the west side. No place like home. If there's questions that you've got, go the extra mile and die. On the dash, got my laces tied. Long road, I don't waste no time. Break rules because fate decides. With the team and we chase the light. I make a move, fall down, shake it off. I hate to lose, bad branch, break it off. No room for negativity, praise and love. Prepare for deep part cause we're taking off. Get the mileage, traveling so far from home. Gathering knowledge, other connections. I put the time in to get everything I ever hoped for. It's a challenge and we all in. It's a better future that we go for. Even though these nights seem long, I know that I am never on my own. Know you won the battle once you've grown. Southside, 10 miles in my rearview mirror. I know what it felt like. My goals only getting clearer. East side to the west side. No place like home. If it's questions that you've got, go the extra mile and die. Long road to the south side, 10 miles in my rearview mirror. I know what it felt like. My goals only getting clearer. East side to the west side. No place like home. If it's questions that you've got, go the extra mile and die. fly by now, don't waste all your time. 'Cause I'll go the extra mile.
to hand over the certificate. It will be here, and then I will run to your side because then I will take pictures. And I will take pictures. Please all rise.
Min Deng. The degree committee here present has assessed the quality of the thesis and of the defense you just gave, and in view of its positive verdict, and taking into account your previous qualifications, of course, the degree committee has decided to grant you the degree of doctor. Your promoter, Professor Olde Damink, is authorized to confer upon you this academic distinction in accordance with Dutch University custom, so I invite the supervisor to take the floor.
Do you promise to work in accordance to the principles of scientific integrity at all times, to be careful and honest, transparent, independent and responsible?
Yes, I promise.
That's the good answer. By the authority vested in us by law and in conformity with the decision of the committee here present, I hereby confer upon you, Min Deng, the degree of doctor, and grant you all the rights attached by custom and law. As evidence of this, I now present you with a degree certificate signed by the rector, the secretary, and the other members of the committee, and affixed with the official seal of the university.
Thank you.
It's always a difficult moment, huh? So it doesn't drop. Careful.
Still on? Yes, it is. So, dear Min, or I should say now, esteemed Dr. Deng, right? It's so nice to stand here today and be the first to congratulate you on this great performance that you showed, not only today, but also in the past years. I'm also congratulating you on behalf of Stephen, of course. And I would like to start by going back to the acknowledgement section of your thesis, because one of the first sentences that you wrote there was, time flies, but memories last forever, right? And I think today, time really flew by, so far at least, and I'm sure there will be really a lot of good memories. I already have them now. I think looking at how you were standing there, you also already have some good memories probably about this ceremony. And you were really shining in the spotlights today, I think, which you normally don't like so much because you're really a modest person who likes to be in the background. But today, it's really no time for you to be in the background. You can be really proud of what you did. At least we are really proud of you. And I think you should really try to enjoy it for a long time to come. So that's for today, but let's first also go back a little bit to where you came from, because it's a long way from here. And actually the first time that we heard from you was back in 2018. And then you sent an email to Hans van Eyck, all the way from Nanchang in China. So actually he did not approach you or me, but he approached Hans van Eyck, who is working in our mass spectrometry lab. And probably that has to do with your own background, because as a master's student you were working on this technique. But I never really discussed with you why you contacted him, but we were lucky that he forwarded your email to us. We were looking at what you wrote, and it was quite clear that you were like a very promising student, because you had already won a master's thesis award. With your work on a very different field, because you studied botany, and actually you described work on detecting pesticides in honey. And you even owned, I don't know if you still own them, 2 patents for this method, which I think is really unusual for a student. So that was already, you know, very impressive to us as well. And we kept that good impression of you when we set up the Skype meeting, because we were still working with Skype. It was pre-corona. We don't even remember the program anymore, but it worked very well. The connection was good. And we agreed basically that you could come based on the impression that you made. But then, of course, the next goal was for you to get some funding to be able to get here. So you applied for the Chinese School Council scholarship. Yeah, this worked out well. And at some point, you emailed us that you could come. And then in September 2018, you arrived in Maastricht. And actually, this was maybe quite difficult for you in the beginning. I think it was a completely different world. And you really needed some time to get used to the new environment here and to overcome the language barrier. And on top of that, actually, your project was not so well defined at the start, right? Mm-hmm. It was a bit unclear. So together with Frank Schaap, we tried out a couple of things, but it was a rather difficult first year, I think. And in which the time flew by and maybe the memories were not so good. And then sometime, I think late in 2018, we sat down together and thought like, okay, we really need to talk and to get a better insight in what the project would be. And only then it was clear that you would be working on a 100% CACHEXIA project. And from that moment, you settled in much better. The things went much better. You are a quick learner. And also a very independent guy, I think, and an original thinker, which you also showed today. And things really started to take off, and together we decided that you would focus on adipose tissue browning and also GDF15 in CACHEXIA. And you quickly mastered all the techniques that you needed for that in the lab, and you were just about to really get some nice data, and then Corona hit the world, and it also hit you quite hard. And many of your projects went on hold, or they were delayed for quite some time. And it was also a difficult time for you personally, when you went back to China, for example, for Chinese New Year, and you had to stay in quarantine basically forever. Just like your wife, and you were separated for a long time. It was a really difficult time for you, right? Mm-hmm. Yeah, and also because of Corona, we did not have a lot of social interactions outside of the lab, unfortunately, and that's really a pity, I think. But still, you left also a lasting memory with my kids, with the way you were shuffling the cards during the board game night that we had. That was really impressive, and they are still talking about that, so they have some good memories about you. And I also have some fond memories still of, for example, our trip to Lisbon, for the CACHEXIA conference, where we really had a good time. And you delivered an excellent presentation there in the Young Investigators Award session. So that was also a really good achievement. You showcased your work on MYOSTEATOSIS and inflammation, just like you did today. And it was also very successful at the time. So Min, you always bring something unexpected. I remember very well that you just accidentally told me, by the way, I got married last week. I was really like, okay, you never told us. It was really a surprise, a nice surprise. So it's quite typical of you. And also, more recently, when you left the lab for your postdoc in Oslo, it was really quite unexpectedly, and you just left from one day to the other. And I'm really happy that today you're back. And we will celebrate that you're back and celebrate your good performance today. And we're looking forward to your next unexpected move or achievement. And maybe this will be you winning a prize for the best science visualizations, because I think you're really great at that. And it was also shown today in the presentation. It was noticed by all the people here, I think, that it was really very educational and a big, good skill of you. But your next unexpected achievement could also be a real cool Nature paper based on public data analysis, because you're also a really true public database wizard, I think, and that's really very important skill today as well. And on the other hand, I think it's also clear that your next unexpected move could be for being the best cat owner in the world, which you definitely are, I think. So, Min, it was a real pleasure to work with you. I'm glad that you will stay in the CACHEXIA field so that we will stay in touch, also scientifically. I have no doubt that you will be very successful, and I wish you all the best in your future career. Thanks a lot for everything. Congratulations again to you and to your wife and the rest of your family, and also to the cats, of course.
Thanks.
Thanks.
Thank you.
And then it's my honor, Dr. Deng, to also on behalf of Maastricht University and the whole academic community here, congratulate you with the degree that you have acquired, and I extend those congratulations to the supervisory team, to your wife, of course, to the family, friends, colleagues here online, to the M4I Institute. I think you're one of the, the persons who really show that this has been worthwhile to start up and now continue the success. And of course, surely the two feline individuals, they have to be congratulated. I'm trying now. Yuan Tang and Q Mai. Is that almost correct? Okay, well, congratulations to them. I found this, I witnessed many defenses, and today I thought was relaxed, but in terms of content, also very high quality. So thank you for that. I would also like to thank the opponents, especially, of course, the two guests here on the screen. As I said before, it's very important to have people from other universities contributing to ceremonies like this, and I think I have to thank you for that. I'd like to thank the audience for being here or online, and that brings me to the end of the ceremony, and I'll just explain how we will do this, because we want to take some photographs here with the two online opponents in the background, which means that I will send the audience out. You can go to the reception area. Some of the people will know where that is. So if you follow them, you will get there, and we'll stay here and take some photographs. And by that, I think I can close this ceremony. Ja, leuk hè? En also supervisors. Somebody should take it. Yeah.
Okay, I will take it.
It can
It's
drop.
yours.
So be careful. Yeah, and then you can show it for the
Also
for the
yours.
picture. Yeah. No, it's nice for